Hi everyone, Dr. Phil here. Hoping this is going to record. So I wanted to just do a quick video. Well, none of my videos are quick. I want to do a video on one of the labs we're doing with replicating the Kirby-Bauer antibiotic testing lab. So what I'm going to do is I'm going to show everyone the McFarlane tube, which will give us the sort of like a visual concentration of the amount of whatever the bacteria is we're testing against or for. So if this was an actual patient and we were really just checking the infected agent, we would be using that as the testing thing. But for right now, I'm going to show you the McFarlane tube. So that is basically what we're saying is the standard concentration for the Kirby-Bauer test. So I'm going to be hoping to get this clear sodium chloride or isotonic solution to be this concentration. The agent I'm going to be using is E. coli, so I made the plate of E. coli. This is a gram-negative bacilli. So generally speaking, it's a little bit harder to kill or inhibit due to the extra cell membrane or cell wall, I'm sorry. So I'm going to be taking some of this off with a sterile swab and putting in here to hopefully get to that turbidity. So we have a relatively standardized concentration. You know, obviously it's not going to be completely standard with everyone because we're doing this visually. The quantitative amount of bacteria is up to interpretation visually. So I'm going to be using my sterile swab. So I showed all of you how to remove that using this end. So when I pull this out, this should be sterile. All right, I'm going to be just taking a very small amount of that E. coli and try to do this off in the camera. And that looks a little bit too turbid for me. So what I'm going to do is I'm just going to add still looks a little turbid. It looks pretty close for this, for all intent and purposes. That's close enough for this lab. So I'm going to leave that in there. That's going to be what I'm going to be inoculating my plate with. Another sterile swab. This is my Mueller-Hinton plate I'm going to be using. So what I'm going to do is I'm going to inoculate my swab here. I'm taking my plate. I'm going to lawn this plate. And by lawning, I'm going to, hopefully this isn't camera. I'm going to be completely covering this dish and change it 90 degrees, cover it again. And then just to be sure, rotate it about 45 degrees. And I'm just watching this end to end. If you want to be really extra, just go around that, place it here. All right, so now this plate should be inoculated with our testing agent of E. coli. Now I'm going to be placing the discs around it. So they'll end up looking sort of like that when we're done. All right, so I have a bunch of antibiotics here. Try to put them in the camera view. Okay, so all of these I labeled the antibiotic and its mode of action. So this is a 50S subunit ribosomal inhibitor. So this is really going to stop the assembly of the amino acids into the protein in the bacteria in the 50S subunit. Right, some of these still 50S. Here's a 30S. So this is actually going to be the smaller part of the ribosome when they come together. This is really going to be the portion that reads the messenger RNA to assemble. This is bacitracin, we probably all heard of. That's a cell wall inhibitor. Ampicillin, it's a beta-lactam inhibitor, so that's going to break up the NAM and NAG units. We have tetracycline, everyone's probably heard of that. Misreads protein synthesis, working on the 30S subunit. Penicillin, everyone hopefully has heard of that. Neomycin, all right, prevents protein synthesis also. So I'm going to just move the discs off camera and just pop a couple on this piece of paper here. I'll put them on here so I can see them. So I'm going to use, I'm just going to grab 6 different, 4. Six. There's really no order for any of these. It really doesn't matter. So now in lab, we'd be doing 2 plates. One of these would be E. coli or gram-negative, and I generally like to use Staph aureus as the other for our gram-positive. But for right now, I'll just do 1. You get the idea. So I'm going to just go in, see, pop that off. So that's 1. And these are labeled. This is TE for tetracycline. This is A for ampicillin. Their concentrations will be on there also. It's already open. This one's going to be bacitracin. Sure. This is streptomycin. This says S10 on it. There's a erythromycin. Chloramphenicol. Okay, so now I'm going to simply place these on my plate, and then I'm going to label all around here which each one of these is. So I'm going to just place that right about there. Make sure you leave enough room for your zone of inhibition if it's effective. I'm going to just tap it in there real light, just so when I flip the plate, it doesn't. I got a huge bubble there. Not my best plate, but it'll work. So weird. These are both labeled the same thing, but I know they come out of different vials. And this one's going to be, normally I wouldn't in a bubble, but it doesn't matter. So I've got my 6 discs on there. Now, normally I would label 1 through 6 and indicate which is here, so I have a key when I go to look at this. But we're also going to number these around the outside, 1, 2, 3, 4, whatever, with antibiotics, so we can test it later. You want to make sure you label your plate. E. coli, the date, and then what you did. So that is how you would set it up. I would incubate that overnight, pull it out, check it the next day, see what, looking for the biggest zone of inhibition. So here's one I did prior, a while ago. So I literally just labeled these, looking for the zone of inhibition. This is an old plate, just for sample, show people. I want to show a couple of these from last night's lab. So lawning isn't perfect on that, but for a first attempt, I'd say it's really good. Right. So in looking, you can see all of these were effective to the zone of inhibition. So we just measure that and see which has the biggest zone of inhibition. So what's happening is the antibiotic is leaving the disc, diffusing out through diffusion and inhibiting the growth of that bacteria. We can see the bacteria growing in the areas where it doesn't come in contact with the antibiotic inhibitor. Here's another one from last night. All right. The key I want to point out, I'm going to actually untape this so we get an idea. There's probably a lot of glare. I'm going to autoclave this anyway, so it doesn't matter. So we can see here there's a super clear zone of inhibition. And I had a student ask me, which was a great question. This one is a little bit murky or cloudy. So what I want to state is, I'm not sure I'm 100% right, but this clear area is probably bactericidal. Right? And this is not a spore former, so it doesn't really matter at this point. But that definitely definitively killed everything. Nothing's regrowing. This looks like we had a zone of inhibition, all right, but the bacteria grew back. So it's just bacteriostatic, kind of slowed it down, but didn't kill it. So if I wanted to test this out, I could take my sterile swab. I could swab around the zone of inhibition. I could replate this. If something grew, I would say that antibiotic was bacterial static. If it didn't grow back, I could say it's bactericidal. But for right now, we pretty much know it's bacteriostatic because it actually grew back. We see the initial zone, but it grew back. Now this area, if I wanted to make sure it was bactericidal or static, I would swab that and plate it in this area of zone of inhibition. If it grew back, I would say it was bacteriostatic. All right. But if it didn't grow back after I swabbed the zone of inhibition, I can be almost 100% sure it's bactericidal. So it killed it all, and there's no living forms, vegetative cells at all. All right, I'm going to end this, hoping it recorded.